and more efficient. But another thing that happened that was somewhat surprising is 2 of the 12 strains took the exact same route. They took the exact same mutational route to getting larger and bigger. Not surprising, is it? If you think about the selective pressures that are there. The selective pressure is the limiting glucose. And so all of these cells are being affected by that pressure. Any mutation that didn't take advantage of the glucose would be lost. If you had a mutation that would allow you to use lactose and it's not in this environment, those cells die. That mutation was no good. So because of the environment, the selection pressures dictated those mutations that would allow the organisms to survive, have the advantage over those that didn't. But the story doesn't end there. At about, was it generation 33,000? I don't know if I needed another graph up there. At about generation 33,000, they noticed that one of the flasks was extremely cloudy. Its optical density, put it in a spectrophotometer and measure how much light is able to pass through it, its optical density was 6 times greater than the other flasks. Now occasionally they would have had contamination occur where another microbe might get in there and grow really well. When that happened, they'd throw that batch out and they'd go back to one of the frozen cultures and restart it. But in this case, it wasn't a contamination. There wasn't another cell in there. They were still all E. coli cells. What had happened is one of the cells had mutated to the ability to be able to use citrate. Now see, E. coli doesn't use citrate. It's a defining characteristic of E. coli. When we do tests in the lab, when we look for citrate, if it's present, we know it's not E. coli, because E. coli is unable to utilize citrate. Can't use it as a carbon source. Yet, here we have E. coli strains using citrate as a carbon source. What had happened, again, looking at the fossil record, is that on the surface of E. coli, I guess I could pull this down here. On the surface, there are those protein channels we talked about. And the thing is that citrate is too large of a molecule to move across this protein channel. And so E. coli cannot utilize citrate because it can't get it across its membrane. Well, the mutations that occurred at around generation 33,000 actually increased the size of those protein channels so that now citrate could be carried across the membrane and into the cell. Once inside the cell, E. coli has all the enzymes necessary to metabolize citrate. If you remember our tricarboxylic acid cycle, one of the molecules in that cycle is citrate. So E. coli can metabolize citrate. The problem is it can't get it into its cells. This mutation at around generation 33,000 allowed E. coli to now utilize citrate. And so now we have a strain of E. coli that doesn't fit the classic definition of what E. coli is. I bring this in because, one, it underscores the importance of genetics and mutation and how it can lead to advantages that the organism can take advantage of. And it's a classic example of evolution, it really is. Any questions?
So that theory is only for the bacteria?
It's not a theory. This is a study that he's carried out for 20 years. It's still going on. This stuff has happened, yeah. He's observed this happen.
That is only applied to bacteria, or is it all for all cells?
Mutations work the same for all cells, for all genes. If you have a gene that codes for a specific enzyme or protein, say insulin, okay, and something happens to that gene, you won't be able to produce insulin. So, same situation. Now, the thing about mutations that are advantageous for us, we'll never see that. Because our generations, are 60 years long, and we'll never live long enough to see a mutation in the human population that we might take advantage of. We are changing. Our population is changing over time. But we have to look at it at a scale that is hard for us to imagine. 1 million years, you really have to think about it in those terms. And that's why this study is so great, because 45,000 bacterial generations is about equal to 1 million human generations, and so he gets to look at change over 1 million years, kind of. Anything else? Okay, so we have an exam on Monday. We're going to be covering chapters 3,